Thank you very much for giving me the opportunity to talk here today. I'm a consultant clinical microbiologist, so that covers everything from diagnosing infections in the laboratory, working out what they're susceptible to, what antibiotics we can give people, but it also involves going around and seeing patients with the really severe infections, so those in their bloodstream, and also trying to work out, in the hospital setting particularly, are people getting the right antibiotics and do they really need those antibiotics? So that's what we call antibiotic stewardship. And so today, in my talk, I want to tell you a little bit more about what antibiotic resistance is from my perspective as a microbiologist, and why people get these antibiotic-resistant infections and the research work that we're doing in the most vulnerable populations, particularly our bone marrow transplant patients, to try and prevent these infections. So it's quite a simple definition, really, for antimicrobial resistance. It is one or more antimicrobial product or an antibiotic, because it does affect tuberculosis and viral infections and things as well, but I'm really going to concentrate on bacterial infections today. So this is antibiotics, and it's resistance of bacteria to one or more antimicrobials, so it just doesn't work when we give it. And if we're talking really about the severe infections with multidrug resistance, this is 3 or more different types of antibiotic that they're resistant to. And I'll show you on the next slide a bit about ways antibiotics work. But we have different classes of antibiotics that target different parts of bacteria. And normally we just need to use one antibiotic. Sometimes we use 2 or 3 antibiotics in combination. But if you've got 3 of these big groups of antibiotics missing, you can see that things really are starting to get a bit more difficult. And like the picture that Ben showed you earlier, sometimes we run out completely. And every antibiotic that I can get to give to a patient, I test it in the laboratory, and we can't find anything, and that's what we call pan resistance. Thankfully, that's very rare. We can still normally find at least one antibiotic, but it's getting difficult in some cases. So antibiotic resistance, Fleming was right. He discovered a product that was made from an environmental penicillium species. It was growing in the environment, and bacteria and fungi have been living together for millions of years in the environment. And so what we've done as humans is really just take products that these bacteria have met for a very long time and put them into a format that we can give them into a tablet or into a vein to fight the bacterial infections in our bodies. So they've got a bit of a head start, really. And antibiotics work in various ways. So they can work on the outside of the cell wall. And so bacteria can block them. They can change what's on the outside of the cell wall that the antibiotics are binding to. They can pump them out. They can stop the antibiotics getting in the first place. And sometimes they can change what's going on inside the cell, which is where some of the other antibiotics will bind and kill the bacteria. And so they've really got the head start on us over us using these antibiotics for really quite a short period of time, comparatively. And then bacteria are also very clever. They can share bits of their DNA, the genetic code that they have inside them. And if they've worked out a way of changing their genetic code to produce something that can break down an antibiotic, they'll share that with other bacteria. And that's via little circles of DNA called plasmids. And there really is a sort of survival of the fittest evolution that goes on. And this can happen within your own body. If you give the wrong antibiotic or not enough of it, people miss doses, or you're on the wrong one, or we don't sort out a big problem like you've got a big abscess that needs draining, then the bacteria that have got this resistance aren't wiped out. You might wipe out all of the rest of them, and a bit like Ben's box of chocolates, you just get the antibiotics coming back. Those are the antibiotic-resistant bacteria. And the bacteria that I study are some of the worst ones that we have. They're resistant to almost all the antibiotics, and they particularly produce something called a carbapenemase. And carbapenems, or meropenem, is one of the antibiotics we have that is the last one really that we can use when bacteria are resistant to pretty much everything else. And these bacteria, we call them CPEs, or carbapenemase producing enterobacterales. So how much of a problem is AMR? So the map here shows you that it's really quite widespread, and there are a lot of countries that don't really report, they haven't got good mechanisms for reporting how much antimicrobial resistance there is. But basically, dark is worse. And in the UK, we do have a recognised problem that is increasing. And so down at the bottom there, you can see that this graph is showing how over the years, antimicrobial resistance has slowly been increasing. And actually, if you look at the map on the bottom of the screen there of the UK, you can see that there are pockets where the darker colours are, and one of those is over London, which is where we work at Imperial. That these are areas, for whatever reason, population density and things like that, that antimicrobial resistance is higher. And so, you know, this is not just somebody else's problem. This is very much our problem that we have to deal with, all of us. And it's very expensive, actually, for healthcare. So all those additional infections that people get in healthcare cost a lot of money. People are blocking beds. They can't get out of hospital because they've got an infection. They've got a problem. Staff can catch infections, particularly if it's a viral infection that people are getting in the healthcare setting. And then they go off sick. And we did a cost estimation, actually, of how much an outbreak of these really resistant CPE infections can cost. And we had a problem on one of our wards, and we closed the wards. It was a surgical ward, and we've published this, so I can tell you that cost us over £1 million. That single outbreak, and that was mainly because we were closing the wards and we couldn't do routine surgery. But it was also things like the amount of time that it took of the staff, particularly our infection control staff, to try and actually get control of this and work out exactly what was happening, all the estates people and the cleaning people that we needed to get in to help fix it. And so what I haven't really touched on is what this actually means for the patients. So unfortunately, in that outbreak, when we look back a year later, these were very vulnerable patients, and more than 50% of them had died within that year. Now, that was really not due to those infections, as some of them it was, but mainly it was because these were very vulnerable patients with kidney disease and vascular disease and cardiac disease, who, when they get an infection, it really is just sometimes the final straw for them. And when we've got antibiotic-resistant infections, we're resorting to really quite old drugs that can have nasty side effects, particularly kidney side effects. And it might mean that for patients, if they've got a resistant infection, we have to delay surgery that can be really important for them, or things like chemotherapy. And if you're not working somewhere that has access to really the modern antibiotics, there can be delays in getting that therapy of sometimes a couple of days while you have to get them in from a central source or from another hospital. And there is the potential that some of these infections, therefore, might be really untreatable, these pan-resistant antibiotic infections. How do we spread it? Sorry, another gory image here for those who are over lunch, but it's quite simple, that a bacteria enters the system somehow, and in this case, we're talking particularly about gut bacteria, and it sits there, probably not causing much of a problem most of the time, but then it can be spread sort of by going to the toilet or with vomiting. And then it can be spread through the environment to another patient, or by hands not being washed onto another patient, and it goes in. And then if that's a really vulnerable person, then that can cause a clinical infection in that person, not just sitting in the gut. And so there are these 5 moments of hand hygiene, which are really important to stop spreading. We use these all the time in the hospital, and our infection control colleagues do a lot of time teaching people the importance of this. But if you have lapses in your hand hygiene, not only can it be spread antibiotic resistance between patients, but it can then spread to cause colonization to an infection if you've got what we call a central line in, or a urinary catheter, things that are breaking down the natural skin barriers. And people have a lapse in their hand hygiene when they're dealing with one of those, then that can cause a bloodstream infection or urine infection or some other infection with the antibiotic resistance. So that then goes from a state where it's just sitting in your gut, not causing problems, to one where actually we're now trying to treat a very difficult to treat infection. So I wanted to tell you what we're doing to try and combat this now, having probably slightly scared you all. And so I want to tell you a success story. And this was a 63-year-old patient who had acute leukemia, and his chemotherapy had been quite successful, and he'd gone into remission, so the cancer wasn't there. But it was decided that to make sure that the cancer didn't come back, he needed a bone marrow transplant. And so you'll see on my slides that come up the letters HSCT, which is hematopoietic stem cell transplant. That means a bone marrow transplantation for this patient from somebody else. And he had had quite a lot of infections whilst he was having his starter chemotherapy, with Clostridium difficile infection that Ben's told you about, various other infections as well. And unfortunately, he picked up one of these highly resistant CPE bacteria when he was on one of his visits to the hospital. And we were really concerned for this patient. He was concerned, what would happen? What would we give him if he developed an infection when he had his bone marrow transplant? We knew that his gut had this bacteria that was very resistant living in it. And chemotherapy is very toxic to the gut. It can cause diarrhea, and it takes away your natural defenses or your white cells, which are so important for fighting infection yourself. So during that period where we gave him the bone marrow transplant, what were we going to do? So we had a little chat as a group, and Ben and I took this to our multidrug, our multidisciplinary team meeting, and we said, can we try giving this chap a fecal transplant or intestinal microbiota transplant? Because we may not be able to get rid of the gram-negative, this CPE, but actually we might at least get rid of his C. diff, and so he might not get quite such severe diarrhea when we give him the chemotherapy. So we gave him some antibiotics to try and suppress as many of the bacteria in his gut as possible, and then we gave him some fecal slurry. And this was about 2 weeks before we did his bone marrow transplant. And this slide, because he was so happy with the outcome, he allowed us to write up his case in the literature. And on the left-hand side, you can see the large number of infections he had and all those R's for resistant. And then we gave him the fecal transplantation, and the bacteria stayed around for a few weeks, but by the time he came in for transplant, he was doing very well. And he had one very sensitive bacteria in his bloodstream when he was at the peak of the transplant, when he had no neutrophils around at all, but otherwise we completely eradicated it. And he had no further resistance, and he actually did really well. He didn't get any of the other usual complications that you can get from bone marrow transplant. So we were really pleased with this time. And shortly after this, there was actually a publication came out showing that the majority of people who are having these bone marrow transplants actually do have a really disturbed gut microbiome. Actually, that is associated with a far worse outcome. And they get overgrowth of different bacteria, particularly ones called enterococcus, and these coliforms. And that if you have a higher diversity, so going back to Ben's box of chocolates, if you've got lots of different slides, different bacteria there, then you do better when you're having a bone marrow transplant. And if we look at in a bit more detail at our patients, the ones that we found that get the infections after their bone marrow transplant, the ones that break through, well, these do tend to be the more antibiotic-resistant bacteria. And so, much like that publication in the New England Journal in 2020, it's suggestive in our own population that we are seeing this GI tract disturbance. So this is a PhD which myself and Ben and Mark all co-supervised, Dr. Roma Ghani, and she studied 20 patients that we offered this intestinal microbiota transplant to. Who were patients who we thought were really at high risk, like our first case that we published. And they were generally people who were having a bone marrow transplant, but we also included renal transplant patients who got kidney transplant who were getting a lot of infections. And we followed them up and collected their samples over 6 months and saw how they did. And we were quite amazed that actually the clinical impact on these patients was huge. And this was actually picked out as one of the publications of the year at the European Infectious Diseases Conference in 2021 as the most hopeful sort of intervention. And we found that they had fewer serious bloodstream infections. They were in hospital less long. They had less antibiotics. And overall, they did much better. And there were a couple of other case series that came out shortly after ours that showed similar sorts of findings. Everyone did them slightly differently and looked at different measures. And then we did a further analysis with 2 of our hematologists to look at actually what happened in a bit more detail in those bone marrow transplant patients of that group. And we found that indeed they had got better much quicker. They'd had far fewer days of fever after they'd had their bone marrow transplant, and their overall survival was much better. So we've now turned that into a randomized controlled trial to see, actually, are we just sort of biasing it with the patients we pick here, or actually are we onto something? And this is a multi-center trial that we've been doing called the MAST study. And it's been run over 6 centers, and we've pretty much finished treatment now. We're just getting the last few samples from the last few patients. And some patients got a placebo, so a fake pill that looked exactly like the one, and other people got the fecal transplant pill. And they don't know which ones they got, we don't know which ones they got, and we're going to be studying the outcomes. And we've published sort of what we're planning on doing and what samples we're collecting. But mainly, these are all patients, and we're doing it as prehabilitation before they have a bone marrow transplant. And we're going to be looking at, have we managed to restore their gut diversity, and then the secondary outcomes. So all I wanted to do really at the end was to just say a huge thank you. I'm so sorry you've missed my thank you slide, because the NHS server logged me out of my presentation. But it was really just a thank you slide to everybody who's participated in this trial, patients, and also all our collaborators at all of the other sites. And we're really optimistic that this might actually, hopefully, in our most vulnerable patients, offer some hope for the future against antibiotic resistance. Thank you.